Hey, everybody. I am going to make an effort here to get you going forward on our first lecture of Chapter 23, which will give a pretty solid overview of things that can affect overall populations so we can get into our conversation of microevolution and the Hardy-Weinberg equilibrium. Before I do that, though, I wanted to just touch on a few things in the announcements page for today. And just make sure that we're all kind of together on the same page and that you guys know my expectations as we move forward so that when we do communicate, we have good conversations about just clarifications of things that are going on, whether it's through the actual work we're doing or what's being assigned, if there's anything that's confusing, because there is a lot here. As I jump into this, as you can see at the top of every day will be the details, that page that will give you everything. That is your number one first go-to every time that I post. As you go down, I have a link already posted for the video created earlier today of me walking through the answers of the reading guide for numbers 1 through 13. Please go through that. It's about a 10-minute video. Like I said, as I finish creating this, there will be a link that will go up that will be a lecture of the PowerPoint you see at the bottom here, the Hardy-Weinberg lecture. As you move on down, you'll see a couple of links for assignments that we need to submit and the due dates that are there. The actual problems that you need to work on for the Hardy-Weinberg assignment are listed right below here. And then finally down below, the discussion thread. As I mentioned a couple times through documentation here, as you guys ask questions here, even if you don't have questions, please guys go into that discussion thread and check in with your classmates. As we work through this, I'm leaning on you guys too to work to make this a collaborative space and not just one where I'm throwing info at you and waiting for you to turn things into me. The more we discuss material, I think it's going to help all of our understanding of the content that's coming at us. Otherwise, joke of the day responses, keep it up, guys. I appreciate hearing from you. I do need a laugh and a smile from you guys, as I would hope that you guys need the same on your end of things. I'm going to hop into the informational page before I jump into the PowerPoint so that I can at least touch on a few things, some announcements that I put at the top. Let me slide this over here. First, great job on your submissions. We had a 97% turn-in rate for the 69 kids that are in DE Bio. Super happy about that, guys. Thank you so much for staying diligent and staying on top of the work that's coming your way. As I say in number 2, please read everything as soon as you can after it posts. Please do not wait until the next day. As time gets tight, Schoology gets busy. I know I had some of you that were struggling to get things to post yesterday afternoon between 2 and 3:25 p.m. I got to assume it's because it's during the day and Schoology is just overrun and overloaded with a lot of students trying to get their things turned in. The sooner you can stay on top of things, the better. And that also helps me because I've already had 1 or 2 questions this morning about the posts for things that are due tomorrow. I'm doing everything I can to work nonstop throughout these days to make sure that this is as seamless a transition for you guys. So I do need your support as you look through things. Please answer every question that you can before you come to me immediately with those questions. Sometimes they won't be answerable and I do need to help, but I need your help in asking that you guys figure things out along the way too. Please work to set a schedule for yourself so that you can get to the posted work sooner rather than later. As I say in number 4, guys, get the Google Hangouts app on your phone. That's not just for me, but we just had a Google Meet with the staff. So all of us are up to speed in using Google Meet/Google Hangouts for quick face-to-face meetings. That will become more important as time goes by. Continue to help each other on the discussion threads. Like I say here, I'm actually willing to give you guys extra credit if you help others that have questions. So if you're not even asking a question, but you see that there's a post on the discussion thread, it should show a little yellow box with a number of posts that have been made. Click into that when you're on my page, and that way you can read the question, and it might very well be something that you can answer, whether it's content related or just navigating the waters of this work. Either way, thanks again for the help that you guys offer to each other. working on down. I think otherwise you guys made sense of things the other day. So I'm going to continue to be as clear as I can as we work down this and stay consistent with bell ringers, joke of the day, and then finally, most importantly, the agenda at the bottom. Some of these may change as we go. I may ask some bell ringers that I want you guys to submit that day via discussion so that we can be more real time with our conversations instead of just submitting at the end of the month. But I will keep you posted with that. Okay, I think that's everything that I really wanted to touch on. Let me actually double check. Hey, guys, I will not be posting work Friday or Monday. Those were designated days off for you guys. So I'm going to stick to that and not give you specific things on those days. But that being said, I will then post something for us tomorrow, Thursday, that will be submissions for next Tuesday. So I'll keep you posted on that, and you will get something from me tomorrow. Otherwise, the conversation has come up in regards to office hours that the teachers are offering to you guys. Folks, I'm available these days basically from 8 until 2. If I'm available and not posting a lecture video or something like that, I'm doing my best to respond as quickly as I can to those questions that need quick responses, as I know you guys are trying to get work done. So again, I'm available basically from 8 to 2, Monday through Friday, and then I'll make myself available from 6 to 8 p.m. in the evenings. If it's outside of those hours, I got to take care of other things in my life too. And so I may not respond quickly, but I'll respond when I'm back to my office hours. If it's urgent, I will see your text and I'll recognize the urgency of a text, and I'll get back to you sooner as those situations may arise. Otherwise, take care of yourselves, guys. I know this is a lot, and you're also trying to take care of your families and other things that may be going on in regards to taking care of siblings that need babysitting, etc. Communicate with me. And if you know me at all, I'm flexible and able to work with you guys, but I do need to hear from you. I don't even see your faces. So I really do appreciate those that have texted me and emailed me these last couple of days. It's great to hear from you. Okay, let's get to it. I will get through as much as I can here. It doesn't give me a timer. I have no idea. I'm sure I've been talking for a while already, but let's get to it. So as we look at the PowerPoint, it is measuring the evolution of populations. And so as we follow through this, let's see if I can do this and go to present mode. I think we can. Yes, here we are. So I'm going to get my face at least out of the way in the corner. If it annoys me, I will close it because I don't need to see my face. Otherwise, I do have my lightsaber. I made sure to grab this and bring it home. So I may grab it from time to time just for my own security blanket as I talk with y'all. So we're measuring the evolution of populations. And again, this is a quantitative approach. It is looking at specific populations and the changes that they go through from generation to generation. Things that cause evolutionary change, the agents of. Sorry for the cutoffs. It should say mutation, not mutatio with an N below. Mutation, I think we have a general understanding of changes that can happen to DNA. Gene flow is really just the movement of species out of a population to go somewhere else and to live somewhere else. If they leave, they take their genes with them, and that changes that gene pool. Non-random mating, when an organism chooses its mate based on certain features, characteristics. The picture of the peacock is shown because the peacock is showing off, displaying for its mate its amazingness. And if an individual of the opposite sex chooses that mate because of certain reasons, that is non-random mating, and that will cause evolutionary change. Genetic drift, that's one that a question has come up a few times about, and that is not due to natural selection, but due to just chance changes in a population. This is something that affects small populations more quickly because averages change much more quickly with smaller populations. I give the example of if there's a genetic disease that affects, say it's a rare disease that affects 1 in 100,000 individuals. That's a really rare disease, so it doesn't show up much in a population. But let's say of that population of maybe 100,000 individuals where there's only one individual that has that disease, let's say that 25 individuals leave that population and go recolonize. Let's say they want to get the heck away from coronavirus and they go find an island in the Pacific that is totally uninhabited. These 25 individuals leave. If one of those 25 is the one person that has that disorder, that genetic disease, now that population is 25 people instead of 100,000. So now the percentage of that disease being prevalent in that population is way higher. That is a huge change to a population. And so that will be a quick change. That is a chance change also. And so that falls under the realm of genetic drift. And finally, selection, that connects up here even to things like non-random mating, but that's our understanding of Darwin and natural selection. So moving forward, some more on these agents of evolutionary change, some definitions as I covered them. Gene flow, that migration that happens. We can call that immigration, individuals that move into a population. And emigration, starting with the letter E, is individuals that leave a population. Let's see, as we move forward here, I have a little bit more detail about that genetic drift here if you want to look through that some more. Again, please post on the discussion thread if you have more questions about genetic drift and trying to understand that. Even the picture of down here below of about 10 beetles. And of the 10, or sorry, 9 of them, 6 of them are brown and 3 of them are green. Just by random chance, this person walks by, accidentally, hopefully, steps on 2 of the green beetles. That changes the genetics of this small population. We now have less green individuals. Selection, I believe we have a pretty good understanding of that, so I'm going to move forward. So now we have to get into the conversation of populations and gene pools and understand these terms. Let's jump back, sorry. I can do this. Here we go. So again, we want to understand the basic term of population. A population is a localized, just within an area, a group of interbreeding individuals. Generally, we just state individuals of the same population, or sorry, the same species. But we want to make clear what we mean when we say species. Individuals of a species are those that can interbreed, and not only interbreed, but also create viable offspring, meaning the next generation of individuals of that species. Down below, the next bullet point, a gene pool is a collection of all the alleles in that population. So thinking our genetics, if we're talking about a certain gene, dominant A and recessive A, capital A, little a, we want to think about that and put all the members of that population, all their alleles. So real quick, if we're talking about a certain trait that perhaps I'm homozygous capital A for, and Brenna is homozygous recessive, she's little a. And let's see, who's somebody from my third period? I can't think off the top of my head. Zyra, let's say that she's a heterozygote, big A, little a. And let's say Ansley is also heterozygous, big A, little a. If the population was just us 4 individuals, we would actually count the number of big A's and little a's amongst the 4 of us. Since we each have 2 alleles for that trait, there would be 8 total alleles in the gene pool for that trait. If we were talking about all 70 of us, all DE Bio students and me, 70 of us in regards to that A gene, there would be 140 total alleles, because I just took the number 70 and multiplied it by 2. There would be 140 total alleles in our population for that trait. So that can bring us down to allele frequency. How common is the dominant A compared to the little a in that population? So again, I'll probably through this refer a little bit as an example to us as a population of DE Bio students and teacher, all 70 of us. Let's see. As we move down, let's just, I don't know what to do with you, Mr. Dupree. So evolution is any change in allele frequencies in a population. So if we go from one generation to the next, and let's say in the first generation, let's say of those 140 alleles for our population of DE Bio, let's say that 100 out of those 140 are a dominant A and 40 are a recessive lowercase a. So 100 out of 140 are dominant A. Well, let's say from one generation to the next, just due to shuffling of alleles that were brought up in number 13 in your reading guide. Let's say through shuffling of alleles, through fertilization and random assortment of chromosomes and crossing over, we actually get a change. And let's say in the next generation, it's not 100 out of 140 are the capital A. Let's say it drops to 80 out of 140. That would be a change in allele frequencies in a population, which would allow us to state, yes, on a microevolution scale, evolution has occurred. So what we need to do then is we need to create a null hypothesis or a null population that is not evolving. This is the Hardy-Weinberg population, a non-evolving population as it states here. Anything that could possibly cause evolutionary change, we remove this from our hypothetical population. So first, we make a super duper large population size so that genetic drift doesn't cause those chance changes to percentages. Think back to the green bugs that were squished. It's easy to change the percentages in that population of 9 bugs. But what if we have 9 million bugs and we squash 2 green bugs? That is not going to have a significant change to the population. Next, there is no migration in this hypothetical population. So there is no individuals leaving or coming into this population. Number 3, there is no mutation. There is no genetic change to our hypothetical population. Picture our hypothetical population being like a control group. The control group is what we're going to make our comparisons to. So if evolution is occurring in a population, we can compare to the control group and say, yep, sure enough, this is changing. This is different from our hypothetical population. Number 4 says that there is no sexual selection. There is no non-random mating. All mating will be completely random. There is no choosing of mates. And finally, there is absolutely no natural selection. Everybody has equal fitness towards their survival. We use that word fit to talk about an individual's ability to survive. In our hypothetical Hardy-Weinberg population, there is no increased fitness or ability of one individual to survive better than another. So we want to keep these 5 components in mind of our hypothetical Hardy-Weinberg population. I'll refer back to them as we go. This hypothetical non-evolving population, this keeps allele frequencies the same from generation to generation. So again, it serves as our model. It is a null hypothesis. It is a control group to compare our actual problems that we'll work on. Natural populations rarely, if ever, really occur in Hardy-Weinberg equilibrium. If things stay exactly the same in a population, if the environment doesn't change, for a short term, you might see a population stay the same in its allele frequencies, but you won't see that last for long. So these are the wonderful gentlemen that are given this credit to for the Hardy-Weinberg equilibrium that allows us to quantitatively study populations from generation to generation to just see if allele frequencies change. So let's walk through a simple approach, and this will hopefully get us going so that you guys can work on Hardy-Weinberg problems 1 and 2 so that you can submit those for tomorrow and I can get a look and see how well you guys are picking up on the basics of how to do. So we're going to shift to a different gene. We're going to say that we're looking at the B gene. And in this case, it's giving us an example of fur coat color in cats. We have the dominant B and the recessive B. The frequency of the dominant allele, we're going to bring a new letter in. We're going to refer to P's and Q's for our problems because we're not always going to talk big B and little b. Sometimes it'll be a different letter as we learned in our genetics chapter. Just to kind of standardize things, whenever we're talking about the dominant allele will use just the letter P. and the recessive will be the letter Q. And it's not just the dominant or the recessive, it's the frequency, how often that allele is found in that population. So the frequency will always be stated as a decimal. So think about it this way. If 75% of the gene pool for the B gene is the capital dominant allele, the frequency, or P, would be 0.75. So we will always state that frequency, our P's and our Q's, in decimals. So the frequency of the two will always add up to 1, which as a percent, that would be 100%. So P plus Q will always equal 1. This will be your first formula to go to. Quick, easy math, but we just got to make sure we're using the right decimals. We're using not as percentages, but as decimals for our frequency of the dominant allele in that gene pool and the frequency of the recessive allele in that gene pool. Sorry, I'm checking my watch. I'm trying to not go crazy long on time. As we go forward, the next formula that we're going to keep an eye on, I'm going to say the formula first and then give a little bit of background up above. The formula for this is in regards to genotypes. Again, if I hop back to the previous slide, this is talking about individual alleles, big B's or little b's, the frequency of one versus the other in a population. Whereas we get to here, we're looking at the genotypes. So going back to my example earlier, say I was a homozygous dominant and Brenna, maybe she is a homozygous recessive. The homozygous dominants, the frequency of them in a population will be P squared. The frequency of the heterozygotes will be this number here, 2 times P times Q. And then finally, the frequency of our homozygous recessives will be Q squared. If we take the frequency of each, thinking that it's a percentage, but we're just writing it as a decimal, if 33% of the population is homozygous dominant and 33% of the population is heterozygous and 33% of the population is homozygous recessive, that 0.33333 plus 0.33333 and so on, plus our 0.3333 of the homozygous recessives, that will add up to 100% of the population always. So this is our second go-to formula. Those will be the only two that we're going to refer to. As you're jotting notes down, as you're going through this, my suggestion, I don't show it here, but I would write next to your P plus Q equals 1 formula, I would head that, parentheses it, something right next to it. This is always going to be in regards to allele frequencies. When we write this formula with P squared and 2PQ and Q squared, give a header or put in parentheses next to it. This is in regards to genotype frequencies when we think about each individual carrying 2 alleles for a certain trait. Okay. So that kind of shows that just in a different way. But the exact same idea of what I just mentioned on the previous 2 slides, to separate in our minds when we look at separate allele frequencies versus actual genotype frequencies. So here's a little practice with this work. So if we look at a population of 100 cats, 84 of them are black and 16 of them are white. So how many are there of each of these genotypes? Well, the quick first thing that we have to note is if we have 84 black cats, just by looking at these cats, if I were to cover up the homozygous dominant and the heterozygous, they look exactly the same. You don't know just by looking at a black cat if it's homozygous dominant or heterozygous, which that's important information for us because we are trying to count the number of big B's versus little b's in a population. The 16 whites, every single one of those, all 16 of them, we know immediately that every single one of them is homozygous recessive or little b, little b. That's important for us also as we move forward. So as we look at this, the first thing we can do with this to kind of figure out our unknown variables, if I think about it that way, to figure out our unknown variables, we already know that all 16 of the white cats are little b, little b. We already know they're homozygous recessive. So the frequency of our homozygous recessives can quickly be calculated. I can go right up here as it shows in the blue box. My Q squared, going back to my formula as I zip backwards, my Q squared is my frequency of homozygous recessives in a population. So we can quickly say Q squared, my little b population, is 16 cats out of the population of 100. So my Q squared is therefore just the simple decimal of 0.16, 16% of the population. So then we can actually really quickly from that, we can figure out the frequency of the recessive allele as a whole by just taking the square root of what we just calculated. Because if we take the square root of Q squared, it gives us Q. And if we take the square root of 0.16, it gives us the frequency of the recessive allele lowercase b. The square root of 0.16 is 0.4. So this 0.4 right here is telling us that 40% of the gene pool is the lowercase b. I think that's all I want to hit there. From that, then we can just quickly go back to our first formula of allele frequencies. If we know Q, it's a quick, easy solve for P. Just take our formula of P plus Q equals 1 and solve for P, since that's our only unknown. We'll take 1 minus Q, 1 minus the 0.4 that we just calculated to get 0.6. On Friday, I will work to have a whiteboard so that I can have right here on the screen that I can just do some handwriting as I go too. If this trips some of us up as we go, I don't need you to have this mastered for Friday, but at least to get a start on this, and then I can help you a little bit more with my next lecture. So what I haven't read down here is that we must assume, here, get out of there. It does say must assume that the population is in Hardy-Weinberg equilibrium so that we can use these formulas. Even though we know that in reality, a population is going to change from generation to generation, this helps us to nail down what those frequencies are in a population. From this, we've now got P and Q. So what I've done right here is plugged in P if we want to know, okay, well then how much of the population should be homozygous dominant? Remember, up above we said 84 are black, but we don't know how many of them would be homozygous dominant or how many are heterozygous. Plug in, so it is a lot of just plugging into these formulas. I plug in that 0.6 into P squared. 0.6 times 0.6 gives us 0.36. Again, knowing from our formulas, I jump back, whoops, right there. P squared is our frequency of the homozygous dominant population. So I can plug in 0.6 squared. That tells me that 36% of the population, which is 36 out of 100, are going to be black. Plug in 2 times 0.6 times 0.4. Do it, guys. Plug it into your calculator. Make sure you see it in front of you too. Make sure you see these numbers. If you don't do it right now as I speak, pause this, take a breather. Make sure you just kind of slow yourself down to at least see the numbers that are just jumping up on your screen. As we plug in 2 times 0.6 for P and 0.4 for Q, we get 48% of this hypothetical cat population are going to be heterozygous. So if we took the 2, 48 plus 36, that's 84 total cats. Hey-oh. Things should add up when we double check our numbers. And again, we already figured out actually at the beginning that 16 out of 100 was our Q squared, or our frequency of our homozygous recessive population. If we take those three numbers and add them together, 0.36 times 0.48 times 0.16, we get 1, or the full 100% of our cat population. So if we look at this, using our equation and sliding right there, if we look at this, we can then, if we do our calculations, if we're given, okay, 74% of the next generation is P squared or homozygous dominant, and only 10% is heterozygous, that still adds up to 84% are cats or black cats and 16% are white. But what we need to do then is solve for P and Q to see if things are staying the same. So we can play with these numbers a little bit and make sure that we're staying in Hardy-Weinberg equilibrium. If we don't stay in Hardy-Weinberg equilibrium, again, going back to the beginnings of this HW conversation, if we don't stay in that equilibrium from generation to generation, the population is evolving. Let's see. Dupree might call it good there. And I think just with those explanations, that probably doesn't answer every question. This is not a quick, easy pickup for everyone if there's anything I've learned over the years. So again, what I want you to do as you look through these, the second something doesn't click for you, pause it, jump back and watch again those 10, 15, 30 seconds to make some sense of. And after you've gone through, jump into just questions 1 and 2 of the HW problems and work on those. As you do your handwriting, please do your best to be clear as you take it a step at a time as I ask you to show your work. As you show your work, that will help me as I take a look at these submissions. That will help me to understand where you're at with your understanding, and that will help me to move forward with my explanations. Post questions on the discussion thread. For those that pick it up and you're like, no, I got it. Inevitably, some will grab onto it and just learn to do a plug and chug approach. If you pick it up quicker, be that helpful person. I do want to give some extra credit to those of you that check the discussion thread and see classmates that are in need of help. Man, sometimes your explanations mean 10 times my explanations. So use your voice and help each other. That means a lot to me, guys. Otherwise, I want to cut this off so that I can, so you can press stop and move on with your work in your day. Other things, guys, I miss you and miss talking to you and seeing you, but at least I'm happy that we get this chance to have these connections. Check in with me. Let me know how you're doing. I'm doing what I can on my end to check in with you guys. If you haven't heard from me individually and you just want something from me, send me a text. I'm on my phone or my computer pretty much the entirety of the day. Again, if I don't get back to you during non-office hours, I'm doing other things. If I don't get back to you during office hours, it might be because I'm doing something like this and I'm avoiding looking at my phone. Just know that I'm thinking about you and hoping that you guys are all doing well. I will be in touch soon. You're going to watch me fumble for about 30 seconds as I figure out how to stop my recording. Let's go here.